Hello students, welcome to F.Y.B.Sc class. Today we are going to discuss about the syllabus of F.Y.B.Sc microbiology course MB122 microbial cultivation and growth. So the title of our course is BSc. So let's have an introduction to the course. Now, you are aware with the microbiology term very well as you are studying this subject from the last semester. So, microbiology is a broad discipline of biology and it mainly encompasses five groups of microorganisms: bacteria, protozoa, algae, fungi, viruses. So, in the microbiology course, we are going to study the interaction of this microorganism with the environment as well as how what are the attachment of this microorganism in the human development and how they will affect the society. Now we are facing so many problems with the COVID-19. So COVID-19 is also a virus. Now we are studying how this microorganism they have their impact on the society. The study of microbiology has its extension in various conventional ways. Now, this microbiology can also be used in advanced fields of biology by using microbes as a study models. So, the branches of microbiology they are remain ever expanding in the field of research, and the branches are mainly spread or they can be categorized as pure and applied sciences. So, although the microorganisms were discovered over 350 years ago, but still a huge diversity yet remains to be explored. The knowledge of this microbiology is become crucial and important for the welfare of the society. So, study of organisms or the microbes it becomes an integral part of education as it play a role in the human progress. So, there is always a continuous demand for the microbiologist as work in different areas like the education, industry, research, etc. So, the career opportunities for the graduate students are available in industry and research equally. The objective of microbiology course is to enrich the student knowledge and train them in the pure microbial sciences as well as to introduce the concepts of application and research in microbiology. The third objective of this course is to inculcate the sense of scientific responsibilities and social and environmental awareness about the microorganism. And the fourth objective is to help students to build up a progressive and successful career. F.Y.B.Sc microbiology course it includes three papers for second semester. So paper 1 paper code is MB121 and the title is Bacteria Cell and Biochemistry. The microbiology paper 2 MB122 paper title is Microbial cultivation and growth. Microbiology paper 3 paper code MB123 and the paper title is Practical course based on theory paper 1 and 2. Now, up till what we have discussed is the brief introduction to the F.Y.B.Sc microbiology course. Now, from onwards, we are going to discuss about the course MB122 Microbial Cultivation and Growth. So let's begin with our paper 2 semester 2 syllabus microbial cultivation and growth. So credit 1 is cultivation of microorganism. The first topic which we are going to discuss is the nutritional requirement and nutritional classification. So this microorganism, as they are also living entities, so they also require the nutrients or different macromolecules as well as micronutrients. So we are going to discuss which kind of the molecules are included under the macro and the micronutrients. In addition to that, depending on their nutritional habit, they can be classified into autotrophs, heterotrophs. So depending on the energy sources as well as carbon sources which they utilize. So different classification systems are available for these microorganisms also. Now, design and preparation of media. So, to cultivate any microorganism, we have to provide them carbon, nitrogen source, growth requirement. So, all these things are included in the design and the preparation of the media. So, when we are discuss about the preparation of the media, what are the common ingredients of that media or the common constituent, and what is their main functions within that media? In addition to that, different types of media are available for the cultivation of microorganisms. So, we are going to discuss all the things. Now, as we have discussed, there is a classification system is available depending on the carbon and the energy source. So, in addition to that, different methods are also used for the cultivation of photosynthetic microorganism, extremophilic microorganism, chemolithotropic bacteria, anaerobic bacteria. Anaerobic bacteria are those bacteria which will grow in absence of oxygen. In addition to that, what are the different methods by using which can cultivate the algae, how we can cultivate the fungus in laboratory, as well as actinomyces and the different methods of cultivation of viruses. Next is the concept of enrichment. Now, when you isolate any microorganism from the soil, you have to increase the number of that organism. That is included under the concept of enrichment. Next one is the pure culture. Now, pure culture is the culture which consists of a single species of that organism, that it should be free from contamination. So, different techniques are there. By using that, you can obtain the pure culture like the stick plate method, pour plate method, spread plate method, how these techniques can be used in to obtain the pure culture. Now, next one is to maintain the bacterial and fungal culture using different techniques. So, within this, once you isolate a particular bacterial or the fungal stem, now what's your aim is to preserve it for the long period of time. Okay, so different techniques are available. By using that, you can maintain your bacterial or fungal culture for the long period of time. So, you can use your culture whenever you require. So it includes different techniques like the subculturing, overlaid with the oil. All are the different methods. In addition to that, different methods are there which we are going to discuss. Now, if you are not interested in isolating a particular kind of the bacterial cell, but you want to work on that bacterial cell, then what you will do, different culture collection centers are there, just like our blood collection centers. So through that culture collection center, you can procure your particular bacterial strain or the fungal strain in which you are interested. So the role of this culture collection center is to identify that particular microbial strain and to give it to them who require. So they support or they will provide these cultures to the industries also. So all these things we are going to discuss in the under the heading culture collection centers and their role. Now, in addition to that, we are also going to discuss about the requirement and the guidelines of National Biodiversity Authority for Culture Collection Center. Now, up till now, what we have discussed is all included under the Unit 1. Now, let's overlook the Credit 2. Credit 2, it includes bacterial growth. So within that we have to study the exponential growth model, that is the kinetics of bacterial growth. Second one is the growth curve. Growth curve, it include different phases like lag phase, log phase, stationary phase and the death phase, and the generation time. Generation means time, means the time required for the bacterial cell to be double. Next one is the dioxy growth. As its name indicates, di means two. If two carbon sources are available, then what will be the growth curve pattern followed by that particular microorganism that we are going to discuss. Now, next one is to measure the bacterial growth, or to enumerate the different method, or to study the different methods of enumeration. So you can go for the microscopic method. Now you are aware with the how to handle or use the microscope. So you can go for the direct microscopic count, or different chambers are available like the Newbor's chamber, Petroff-Hosser chambers. Yeah, by using these chambers also you can count the number of cells. Next one is the plate count method, that is the total viable count. Viable means living organism. So you can count the living microorganism. Next one is the turbidometric method. So by using the nephelometry, you can study the number of microorganisms which are present within that solution. In addition to that, to estimate the biomass, you can go for the dry mass as well as packed cell volume study. By estimating the cell carbon and the nitrogen, you can calculate or enumerate the microorganism by using chemical method. Next one is the different factors which affect the bacterial growth. We have to study all these factors. pH. pH means a hydrogen ion concentration. So what is the optimum pH for the growth of, at which the growth of that organism will take place? What happen if we increase or decrease the pH value? How it will affect the bacterial growth? In addition to pH, temperature also influences, so its influence on the bacterial growth. So this bacteria, they require optimum temperature for their growth. So what happen if you expose them to the higher or lower temperature level? How it will affect the bacterial growth? In addition to that, solute concentration, that is the percentage of salt and sugar, how they will affect the growth rate of microorganism. In addition to that, we have discussed the heavy metals in sterilization or disinfection process. So this heavy metal, they also influences the microbial growth. So we have to study this factor and how they will show their impact on the growth of bacteria. References: you can visit Unipune website for the reference. Thank you.